Okay, so good afternoon and welcome all to our webinar today. So our session today is going to be focused on the advanced therapeutic space and is going to touch upon the regulatory routes of bringing these products to market and how this impacts achievement of different value inflection points, which then of course ties into securing funding and investment. So I'm just going to admit some more people, thank you. So having been on the prep call with our speakers today, the discussion went so well and we could have got on for much longer than an hour. So I'm delighted to have the experts lined up that we do today to share with us their knowledge and expertise on this subject area. So just also to highlight that the webinar is in collaboration with PharmaLex, who are supporting our session today. So moving on to the format, you can see I've got that up on the screen at the moment. So after a short welcome from myself, we're going to move on to a presentation from Kate Corman, who is Director and Principal Consultant at PharmaLex. And then following that, we're going to bring in Julian Hitchcock of Counsel at Bristow's and Dominic Schmidt, Partner at Syncona, to have a discussion, and Kate will also come into that as well. So we've got a really exciting session planned. Can I just ask that you utilise the chat box to submit your questions, and then we're going to reserve time at the end of the session to take those. Please don't be shy. You don't have to hold on to them. You can drop them in throughout the session, and I'll make sure to pick those up. So if you'd rather, you can message me privately, and I can then pass this on to the speakers at the end, or you can just drop them into the chat box for everyone to see. And so to highlight also, we are recording our session today. So with that, I'll hand over to our first speaker, who's Kate Corman. So Kate has extensive experience of working in quality roles in the pharma industry and is also a practicing QP, a qualified lead auditor, and in SME risk management. So Kate, I'm going to stop sharing my screen and then I'll let you share.
Thanks, Alicia. Perfect.
Thank you. Oh, sorry, just one more thing. Just if you're not presenting or speaking, if you can just mute and turn your camera off. Thank you.
Okay, so just one moment. Okay, good afternoon, everybody. My name is Kate Coleman, as Alicia said. I'm Principal Consultant with PharmaLex in Ireland, working mainly in quality management and compliance. But I also work across our network with lots of different offices within the PharmaLex network around regulatory issues also. So there's a picture that looks completely unlike me thanks to COVID-19 and lockdown, but that's how you might see me if you're looking me up on LinkedIn. So to start us off, what do we mean when we refer to value inflection points for ATMPs? So obviously, they're very innovative products that attract a lot of investment interest, but at what point do you want to get involved, or what are the things that you're looking for if you were looking to get involved? So we have all of our different stages. So our preclinical studies, does this product have potential? What's the associated toxicity with it? What's the the dose that may be applicable to it when it gets through the different phases? Going into phase 1, then this is the point at which we find out, is it safe? Is it something that is a runner or not? Is it going to work? Is it going to be safe for humans? Phase 2, then we're looking at, is it effective? Does it actually have activity against the indication that it's proposed for? Phase 3, then, so how does it compare with other items on the market? Does it have any similar types of drugs that are out there being used, or is it something completely novel that is, giving hope to patients that maybe didn't have any treatment routes in the past? And how easy is it going to be to get a marketing authorization for it as well? Is it something that the regulatory bodies are going to be happy to approve? And then when we get into commercial, then it's really about, how do we meet demand? Are we going to be able to scale up our operations? Are we going to be able to provide robust manufacturing platforms? And when we talk about value inflection, then throughout this process, we're really looking at the points where, if we invest in something along this road, at what point can we be sure that we're going to be able to get this to commercial and we'll have a return on investment? And risk and quality aspects are very important in this as well. So, we want to make sure when we get down to commercial and particularly when we get to marketing authorization, that we have all the data that we need in order to satisfy the regulatory authorities that it is safe and effective, that it does, provide a clinical advantage for patients that are going to be getting this product. And that from a commercial perspective, it might be a great idea, but it's no good to a patient if we can't get it to them or we can't manufacture it in a robust manner and meet the demand that there is for it. So I suppose that's really what we're looking at in terms of the pathway. So innovation to commercialization then. So obviously we've got investment in these types of products, and we've got drug discovery where we've research innovation and research funding. And then when we move into the preclinical and clinical studies and regulatory approval, that's really where we start to see the need for investment. So, clinical trial design, manufacturing of IMPs, oversight of clinical trials, if the sponsor needs a lot of support in that situation, we've data analysis and interpretation, compilation then of data into our MAA or our NDAs or INDs, and trial funding as well. So this is really, and I suppose anybody who's had conversations around pharma in general, not just ATMPs, this is often an area that kind of gets forgotten about, that the cost of all of this to actually get a product to the market, it can be very significant. Even thinking about all the different contract manufacturing organizations or contract regulatory organizations that may be required, there's fees and costs associated with all of them. And when we talk about ATMPs and cell and gene therapies, as they're known in the US, really it is the kind of innovation and that we talk about, and particularly patient populations, when something, is maybe going to come onto the market, or it's in research, or it's in trials. There's a lack of understanding sometimes about what if this doesn't get to commercial, or the reasons why it might not get to commercialization. And that can lead to frustration as well, as to why these things can't be made available earlier. So an understanding of all of the input and investment that's needed in these aspects prior to commercialization is important. And then once we get our marketing authorization, then we're going into commercial. But before we get that marketing authorization, we have to have some idea of how we're going to actually manufacture this. What are the capital costs that we will need? Particularly for some of these therapies that are, there's very specific challenges with the manufacturing of them. And sometimes, we can do what we need to do from a bench sort of scale, but getting a scalable platform for some of these therapies is difficult. Getting reliability of yield and having, innovation in terms of novel manufacturing platforms. And it can go from, one extent to the other, or one extreme to the other. Some ATMPs can be manufactured in, a simple enough environment where we have, unidirectional airflow protection within a grade C background, and it's nothing too complicated, to then going to the other end of the scale where, we're struggle to find a robust manufacturing method that you can scale up for these types of products. And all of this then needs capital investment. Even if a company or, an SME has a small operation and wants to do this virtually, they still need to enter into contracts with CMOs, they need to do audits, they need to get QP declarations, they need to figure out the supply chain, they may have to, work with 3PLs. There's a lot of, again, behind the scenes that people maybe don't often see. And after all of that, as people who may invest in these types of innovative therapies, there is a requirement to have return on investment. Although from a quality point of view, and as a quality person myself, the patient is always center, and giving a patient a therapy that, gives them hope, gives them the chance maybe of a cure or being able to manage symptoms that they couldn't manage before. That is the priority. But at the end of the day, when you've got investment in, you need return on that investment in order for that cycle to begin again. And therefore, risk reduction is really key throughout the entire process. And what we'll talk about is, some of the ways and some of the things that we consider in terms of risk reduction. So when, in the research and innovation phase, we have to make sure that, we don't dampen innovation with practicality so that you don't have the commercial aspects coming in too soon. But at the same time, you need to have a strategy through the life cycle to make sure that you're mitigating risk where you can, and that you're putting in measures that try and help you get through the process with the least amount of pain and least amount of cost, and in a timely manner as well. And luckily enough, the regulatory bodies, they've risen to this challenge to a certain extent. There's a lot of way to go, but they have brought in new ways of doing things to help this. So ATMPs, there are very unique challenges associated with them. And when we talk about ATMPs, and it's a very European term, obviously they're cell and tissue-based therapies in the US. They're a class of medicinal products that represent, this was a shift away from the traditional medicinal products, but they offer the potential to cure previously uncurable diseases or give patients a longer and higher quality of life. One of the first ATMPs I would have become aware of was the CAR T cell therapies, which really gave such an amount of hope to people who had, were in the end stage of things like leukemia, etc. And with, when therapy went well, suddenly they were going into remission much quicker than they were expected. And the stories associated with them really caught my attention. But also with that, the challenges, and as a QP, the challenges of actually getting these to market and having them follow a pathway that's been designed for your more typical high volume, large patient population medicinal product, that provides a challenge that we need to deal with. So as I said, ATMP, it's a very European word, but they're cell and gene therapies in the US. And the key challenges then are, first of all, personalized medicine falls under ATMPs. And using therapies that, are specific to a patient, we need to think of how do you actually get a return on investment for something like that? What is the cost associated with it, and what is an appropriate cost to get in return, or appropriate charge to get in return for it? So there's a key commercial challenge here that is new enough in relative terms to the industry. I suppose ATMPs as well, there's a current lack of understanding in the wider community, not so much in the pharmaceutical community, but within, the wider population, where patients that maybe have diseases hear of these things coming on the market, and then it goes away and they don't know why. They don't know whether it just didn't get through trials, was it safe? Or, sometimes it's just not commercially viable, but would represent a cure or, a good therapy, but we just can't, we can't just make it for free. And then there's also the part of cost as well, the cost to the patient, what's reasonable. And obviously, if you're somebody who's ill, you're not going to understand why you have to maybe pay a premium for something like this. So that lack of understanding can challenge the whole situation with ATMPs. Sometimes they are expensive to manufacture, and it's not just about manufacturing equipment. It's not just about trying to define a manufacturing process and all the capital investment that entails, but also they are very unique types of manufacturing, and they require more expertise and therefore higher qualified individuals to run these processes. They're still quite unique, so that experience is unique, and therefore that comes at a premium as well. So it's not just the capital investment, it's also the manpower required to be able to do this well. Small patient populations obviously mean, return on investment is more difficult, it's higher costs. And if something has orphan drug designation, there's only a very small patient population that is available or going to need this therapy. So how do we overcome that? Some of these therapies can be used for multiple indications. So how do you manage that in the product life cycle? Like any type of biological drug and anything where you're relying on biology, there's variability. As a microbiologist, I've seen this right through my career, that it's not like chemistry where we have a defined specification that's very easy to see whether it's black or white, but you do get batch variability. And where you're using particularly cell and gene therapies, you know, where you're looking at donors potentially, or you're taking cells or blood products from a singular patient, there's going to be obvious variability. And with that variability, then it begs the question of then how do we set up a strategy for how this is released? How do we have a strategy for how we get the data to bring it to market and for our marketing authorizations or INDs? And with that variability, then that's where you lead into regulatory complexity. So a little bit of, you know, all of these things where it's small patient population, it might be for an indication that's not well understood, maybe personalized medicine. There's a lack of understanding even in the regulators that are involved with this. There is an inherent batch variability. There's a difficulty in trying to, you know, come up with a manufacturing process that's robust. And all of that then adds to the regulatory complexity. So where do they fit within the regulatory pathway? And then QP certification challenges as well. So a lot of these therapies, they really do challenge our current guidelines and our current thinking in terms of how a drug gets to a patient. So is a QP or qualified person, for anybody who's not familiar with the term, and that's the person who certifies a batch or a product for release before it's given to the patient, you know, is that person going to be at the bedside now? If something is made up in a hospital under hospital exemption, how does that get released? And in a standard process, where there is a standard process, can the QP expect to maybe not have the same type of test results that they would have previously based their release on or their certification on? So it brings up all of these questions in terms of how we manage all of this. And then there's a balance, you know, so we want to meet patient needs. We want innovative medicine. We want people to be healthy. But we also want the product to be safe and to be of, you know, an appropriate level of quality. But then we need to consider the return on investment and commercial success. And I remember having a discussion with a manufacturer who deals with ATMPs and cell and gene therapies. And one of the instances that they found, you know, so unusual, having been in the industry for a long time, was with this ATMP, that essentially the patient who was going to be receiving the therapy was, you know, had a low percentage rate of survival, and they didn't have very long. So, and they had a contamination in the ATMP during manufacturing because it was a very manual process. But in a normal scenario with a medicinal product, you know, that batch just wouldn't be released and the patient wouldn't get the therapy. But where ATMPs bring up these questions is that in that instance, you know, the clinician was brought in with the QP, and a risk benefit scenario was looked at in terms of, if the patient doesn't get this therapy because it's contaminated, then, you know, it's almost certain that they wouldn't have very long. But if they were given the therapy with an adjunct antibiotic therapy, then, you know, the chances are that there may be a success and they may have a longer and better quality of life. So these are the types of balances that ATMPs and cell and gene therapies really bring to the fore. It's that risk benefit and also the commercial success versus meeting patient needs and innovation. And somewhere we need to find the balance there of what we invest in and what we maybe don't. So they do, ATMPs really do, they are innovative, and they represent, you know, a paradigm shift in healthcare and how we consider healthcare. And when we look far into the future, a lot of people like to think about the fact that we will be moving away from this one standard medicine for a large population of people for a specific indication, whereas now it'll be, you know, or in the future, it'll be much more tailored to both your genetics, you know, your predisposition to a certain condition, and to really looking at what's making us ill and how do we make it very personalized. So their unique characteristics and challenges really drive the necessity for a strategic plan of risk reduction throughout the product life cycle, to ease that financial burden associated with the regulatory process and bring the product from the bench to the market. And there are lots of different pathways that can be used to get these types of products to the market. And like anything, you know, it seems very, very complicated, but having a strategic plan and breaking it down step by step, we won't go through it in that granularity today, but hopefully we'll give you something to think about. having that plan, once we've found something that has the potential to be an innovator, and we've found something that really could work, even if we're at the preclinical stages, it's looking at what are the risks associated with that, doing the due diligence, making sure that we're capturing data all the way through, and that we're really challenging that data in terms of risk. And it can't be stressed enough, I suppose, of having people who are experts in this area kind of bring you through it can be of huge benefit as well. So as I said, risk reduction really is key. So to reduce the risk of failure at every point in the process or in the product life cycle, from research and development to commercialization, and then reducing the risk in regulatory approval. And we do have some pathways that I think really do give access to the regulators, the rapporteurs, and to people in scientific advice, that there is situations now available where we can get advice and where we can really bring the regulators on the journey from the much earlier in the process. And then this optimizes the available regulatory pathways to us for approval, and all of that then helps us get the product to the market in a much more reasonable time frame. And the sooner you get it to market, obviously time is money, the lower the costs. So just to start off with some of these pathways, one that I suppose was interesting to me was the hospital exemption. And there's been a lot of debate, I suppose, in the industry around hospital exemption. So the hospital exemption is particularly in the EU, and it follows the implementation of the ATMP regulation a long time ago now, it seems 13 years ago, by the EMA. And it's at the same time became mandatory for ATMPs to follow the centralized procedure to obtain a marketing authorization. So you couldn't follow any other pathway but the centralized procedure. And it requires that ATMPs must fulfill the same high regulatory standards as other pharmaceuticals, including manufacturing compliance with GMP, having the pivotal clinical trials, pediatric investigation plans, and all those things that go with a normal medicinal product. Because of the nature of ATMPs, this did put a really significant burden on ATMP developers and manufacturers. And as we all know, these are mainly represented by SMEs and academic institutions, and not so much by, what would traditionally be spoken about as big pharma. And some of these companies only have limited regulatory expertise, budget, and personnel. So they have all of the innovation and all of the ideas and the initial research, but sometimes they just don't have the tools or the backup or the support to actually then get those types of products onto the market. And particularly as a consultancy company, and any consultancy company, these are the areas that, we've seen grow over the last number of years of providing that type of support. So an exemption from the centralized procedure then was introduced to allow marketing of certain ATMPs in individual EU member states. And this was known as the hospital exemption. So it permitted the use of ATMPs in their respective territories without the need for marketing authorization. And it can only be applied for custom-made ATMPs used in a hospital setting for a specific patient under the responsibility of a physician, and only for use within the member state where it is produced. So the requirements around this are quite restrictive. And just to compare it with a standard regulatory pathway, and rather than bombard you with lots of text on a screen, I don't want to give anybody death by PowerPoint. I tried to kind of show this in a graphical way. So when we talk about hospital exemption, there is the possibility that when you apply for this exemption and an ATMP is used in that clinical setting, that the results and the data from that can actually be used to start your data collection. But there's a challenge in here as well, that it has, generally these are made in the hospital setting or the clinical setting. They're made, as it was said, for a specific patient. It doesn't really give you an idea of commercialization or, how to scale this up into a more standard manufacturing environment. And does it give you enough clinical data? At the end, you may get data out of your hospital exemption data, but is it going to be enough? Or are you still going to have to do additional regulatory studies in order to meet the requirements for approval? Whereas, you know, if you have a standard manufacturing process built up and you've had the resources to develop that and to put it in place, and you've had the resources to get all your clinical trials, if it's possible to get as much data as possible through your trials, you know, using the centralized procedure, then you may get approval more quickly. And I suppose that's the challenge that you can see there on the top, you know, that the route to market through hospital exemption, although it's a good thing for patients that need the drug quickly, it does add in additional steps, and it shows the difference between companies maybe that have support and those that don't in terms of timeline to market. And obviously the one at the bottom there is potentially going to get to market quicker than the one on the top. So which is the most efficient path? And efficiency, again, you know, means a lot in terms of being able to get these products on the market, get a return on investment, and make sure that they're not just, you know, spending a long time in that regulatory process. So with that in mind, looking at the EU regulatory landscape then, so there's multiple pathways. You've got accelerated assessment. You've got authorization under exceptional circumstances. So where you've got a shortage of data or data cannot be obtained. So for example, for any of these more personalized ATMPs where it's difficult to get the type of data that would be required in a normal application. So like rare disease studies with small patient populations, you can get an authorization under exceptional circumstances. And it allows for ongoing safety monitoring after a drug is on the market with an annual risk benefit assessment, reassessment. So again, it's back to this conversation that I previously had with a clinician about the risk benefit scenario and balance. So what are the risks to these patients and what is the benefit that they can get from having access to this drug under a more expedited pathway for approval? Adaptive pathways licensing then, so this is used when more data is needed to widen a drug's indications. So the focus is on early dialogue, again, between sponsors and regulatory agencies, as well as an iterative development approach that leverages then the real world data. So really the key here, and this will come up time and time again, is about speaking in advance with the regulatory authorities, letting them know what you have in the pipeline, letting them know what the patient populations are, what you think the benefit of this will be to your patients, and starting that dialogue so they can start to understand, and particularly if you're looking for an expedited approval, that they're with you on the journey of that and you're not landing it with them at the end. And the iterative development approach as well, you know, means that the data that you're using, it is real world data, and you can widen the indication for the drug, which again leads to another value inflection point if you get this, because obviously it makes the drug more commercial. Conditional marketing authorizations then, so that offers a temporary 1-year approval in situations where the benefit of immediate drug availability outweighs the risk of less comprehensive data than normal being available. It's allowed when it's likely that comprehensive data eventually will be gathered. And that's, I suppose, the difference between authorization under exceptional circumstances and the conditional marketing authorization is that authorization under exceptional circumstances does not assume that the full data set is eventually going to be gathered. It is for those types of ATMPs or cell and gene therapies that, you know, may never have the type of data collected for a normal application, which would be expected in a conditional marketing authorization. The conditional marketing authorization just makes sure that patients get, you know, an earlier access to the drug because the benefit really outweighs the risk. But you still need to pull together that data set eventually. And then this brings us on to EU PRIME, and the EU Expedited Pathway for Priority Medicines is known as PRIME. It's an accelerated assessment based on scientific advice. And what they say on the EMA website here is, as their explanation for PRIME, is that by engaging with medicine developers early on, PRIME is aimed at improving clinical trial designs so that the data generated is suitable for evaluating a marketing authorization application. Early dialogue and scientific advice also ensure that patients only participate in trials designed to provide the data necessary for an application, making the best use of limited resources. So getting PRIME designation really is, in Europe, is really something that can help you along the pathway to getting regulatory approval. And I'm involved with a client project at the moment with our regulatory colleagues in terms of PRIME. And the part that we emphasize in that is really giving the information up front, making key decisions, and going through what you plan to do and looking at where the risks are and trying to come up with mitigations to those risks. And even things like looking at down the line, you know, if your drug comes to market, do you have a product lifecycle management document like as per ICH Q12? Have you defined your established conditions? So can you save maybe on, you know, variations after approval to see can you fit into that ICH pathway for, you know, only needing to go again through a variation approval cycle when you really need to. And it's using a risk-based approach so that your regulatory burden right through your process using PRIME and then using something like ICH Q12 after commercialization, that you're always thinking about reducing that timeline, reducing the cost, and getting your product to patients as quickly as possible. Products are eligible for PRIME if they offer major therapeutic advantage over an existing treatment or existing treatment options, or if they benefit patients without treatment options. So a lot of ATMPs or biological drugs that, you know, look at enzyme replacement disorders and things like that, a lot of oncology drugs, but anything that is going to really make a difference to the outcome for a patient over the current treatment, or, you know, if they don't have any treatment options at all, then they would be subject to approval for PRIME. And I just had a quick look there this week, and there's currently 65 products in the PRIME pathway. So there's quite a few that get approved through that pathway as well. And really the key to it is you have to submit documentation going through all of the information around your drug, and you interact early with scientific advice so that essentially they understand what you're trying to do and how you're trying to do it and how you're trying to get that data set together before you ever go for your MA. So it's a very valuable pathway for drug developers and medicine developers. On the US side of things then, the US regulatory landscape also has a lot of different pathways that can be used. And particularly over 2019 and 2020, the FDA have published a lot of advice and a lot of information in terms of what their position is on cell and gene therapies, as they refer to them. And Scott Gottlieb from the FDA around BLAs, you know, discussed to advance their goal of supporting cell and gene therapies, that one guidance that they intend to issue will outline a proposed innovative trial design by which individual researchers can pool their clinical data after following a common manufacturing protocol, and thereby develop a more robust data set for the purposes of gaining a BLA. And what this is really referring to is trying to find ways to help clinical researchers to actually get that clinical data and pool it and make it more efficient so that there isn't this huge regulatory burden on each SME or on a researcher to try and get that data themselves. We've got RMAT, which is the Regenerative Medicine Advanced Therapy designation and the associated accelerated approval with that. So if the drug is a regenerative medicinal therapy or medicine therapy, which is defined as a cell therapy, a therapeutic tissue engineering product, human cell and tissue product, or any combination product using such therapies or products, except for those under the regulation of section 361. And the drug must be intended to treat, modify, reverse, or cure a serious or life-threatening disease or condition. And preliminary clinical evidence must indicate that the drug has the potential to address unmet medical needs for such a disease or a condition. So that is another pathway that can be used for some cell and gene therapies to actually allow an expedited approval. You've also got accelerated approvals for that with a surrogate endpoint. So where an endpoint surrogate is selected, which is proven to be an indicator of the outcome of a trial, and you're allowed then to have accelerated approval under that basis. and breakthrough therapy designation as well, which also offers accelerated approval. And that is what it says it is. It's for any breakthrough therapies that are seen to have a significant advantage for patients. Again, if they're designated as this, they can benefit from accelerated approval and other supports from the FDA. And going back to what Scott Gottlieb said in his address on, I think from January 2019, the FDA said they believe that these cell-based and gene therapy technologies hold tremendous promise for addressing some of the most intractable diseases, but their novelty also comes with new uncertainties and some unique and theoretical risks. So our efforts are aimed at helping innovators proactively address these potential risks while we outline a modern and efficient pathway for the continued development of these innovations. So really, again, it's talking about interacting with the regulatory agencies upfront, keeping them, informed of what you're planning to do or how you're planning to get your data together, how you're planning to submit your authorization or your IND, and what are the things that you're thinking of doing? Are you thinking of using, as they say, ICH Q12? Are you thinking of sharing some of your resources with to get data or to use data from other sources? So all of these things need to be put into a strategy really at the outset, and to use those different avenues that you have to actually get good advice and to show that you're reducing risk all the way through the process. And really that's something that probably is not focused on enough, is actually in each stage saying, what are the risks here? What are the risks that this won't get to the next stage? Or what are the risks associated with the product type that may prevent me from being able to get data to convince a regulator to give me marketing authorization? By taking that sort of approach, it means that for any question that you're asked through the regulatory process, that you'll have an answer and you'll have a mitigation thought through. The FDA also said that our new guidance will recommend parameters for how innovators can introduce advances in manufacturing that promote the more efficient development and application of CAR-T. And in this instance, they were specifically talking about this therapy, without necessarily requiring costly new clinical investigations. So we intend to propose ways to help ensure the safety and effectiveness of the resulting products through available technologies and examinations, and when limited clinical bridging studies may be needed prior to approval of a change, possibly followed by the submission of additional clinical information supplied by real-world data after the change is introduced. So as the whole industry moves towards this new paradigm of risk management, what they're saying here is that with ATMPs or cell and gene therapies, they are also going to use risk and risk mitigation and risk management as a way to make sure that these products can get on the market in circumstances where the investigators or the sponsors have really thought through those risks and have brought all of the information to the table. So the key takeaways then from all of these different regulatory pathways is that reduction of risk throughout the journey from discovery to approval is really, it's a really key area. And writing down those risks, being aware of them, sharing them with all the stakeholders, and working together with the industry experts and industry representatives to help you reduce those risks. Also, communication with regulatory authorities and use of the available pathways, knowing what pathways you could potentially use, sitting down with the regulatory authorities to see which one is most appropriate, making sure you go with answers and data so that when you have these meetings that it's not an open question to them of what can I do, that you've done the research, you understand your product, you have a thought around your proposed process of how you're going to get this product to the market, how are you going to manufacture it, how are you going to scale it up, how are you going to make sure that you meet patient demand and won't have, stock outages. All of these things need to be thought of in advance. And this all saves money and time. And although sometimes we separate the innovation from the commercial aspects, they actually do, all tie together to give that one picture of how this is going to get to a patient and how it will continue to be given to a patient over time and continue to be available. So having a strategy is key, and having the product lifecycle document can really help with that as well. to help you ask those questions that you need to ask and to find the answers and get them documented. And one key thing as well is if you're a sponsor or an SME, or you don't have a lot of regulatory expertise, or you don't have the operational expertise or quality expertise, you know, there's lots of professional help out there. There's lots of companies that have done this before and have gone through these different pathways, and they've gone through any type of issue that can be experienced. So getting professional help is always a good investment. It saves time and money, and it saves mistakes in the long run. So that's the end of my presentation. I hope in that whirlwind chat that it was useful. And as Alicia said, we'll take questions at the end. Thank you very much.
Thank you very much indeed, Kate, for highlighting sort of how unique this area is in terms of sort of the regulatory pathways. I would now like to invite Julian and Dominic to come into the conversation. So the idea is they're going to help to tackle the investment perspective as well. So if I may, just for those who were not familiar, as you may be already, a quick intro. So Julian has extensive experience in regulatory issues relating to emerging biomedical products, services and research to a wide variety of clients at Bristow's. His particular area of expertise includes regenerative medicines and genomics. And Dominic has a PhD from the Department of Oncology at the University of Cambridge, and his research has also been awarded prizes and published in Cell, Nature and Science, and is now a partner of Syncona Investment Management Limited. So I think because of timing, if we sort of go for a sort of a 10-minute discussion, and then I do have one question come through, so I can kind of bring that into your discussion also. So over to you guys. Thank you.
So I just lead into this. So very briefly, what I'd like to do is to set Dom up actually by trying to set out what the regulatory regime is and how it differs. And I'm not going to try, and thankfully, Kate has covered a lot of those areas. My background is actually very related to this. At one point, I used to run what we call the ABC meetings in Cambridge, which are the academic business clinical meetings when I ran the stem cell network here in Cambridge. Anyway, what are they? I think we've talked through what sort of products are: somatic cell products, tissue engineered products, gene therapy products, and the combined ones, which is like a device and an ATMP. And they're regulated, as I think we've seen, under the Medicinal Products Directive. I'm ignoring Brexit for now. There's reasons which we'll come to on that. And that's modified by the ATMP regulation, which we always hear about. But the key thing here then is going to be about the return on investment. And there's no return until you can get the product available lawfully. So which assumes an MA and reimbursement, but I'm not dealing with reimbursement. So MAs. So the MA is essential. And just generally speaking, what I think we've gone over the basics of that. So physicochemical, preclinical tests, clinical tests, pharmacovigilance, risk management, and so on, just those things. But of those in particular, we focus in on the demands placed in relation to clinical trials, but also about the earlier side, the non-clinical early tests. But if we look now quickly then at the clinical trial regulation, why it's different? Well, because you have to comply with standards of good clinical practice, GCP. And that means that, that you tend to come in rather later, basically. The very nature of the beast is such that you're very unlikely ever to administer to healthy volunteers. So no phase 1 and animal models are obviously limited value, and it may never be there actually. So it's going to, when the IMP, the investigational medicine product, let's call it that, but it's a version, is administered, it's likely to be in a 2A, proof of concept study, pre-dose bindings, probably the earliest you'll get. And as Kate said, the dose numbers may be very small. So if we look at Zolgensma, which is for spinal muscular atrophy, which is released on a conditional, like that 1-year exceptional reviewable marketing authorization, the number of patients in the trial was 22. That's not untypical given the diseases which these products tend to be focused on. Anyway, the products themselves are also a bit different. And again, why? They have a short shelf life. The mode of application is different from conventional products. The use of placebos and controls is a bit different. Can you imagine the placebo of certain procedures? Anyway, it's not feasible to generate relevant non-clinical data. We've said if it's tested, that makes a difference. And then there are specific characteristics which require adaptation, sample retention, traceability, and so on. And in particular, in relation to good clinical practice, GCP, the European Commission provides guidance. In fact, the guidance, the most recent is from 2019. It's the first real shot of some guidance as to what GCP requirements there are. And these are really detailed. And I'm not going to give you all the detail. I'm going to spare you that. I deliberately decided not to give you death by PowerPoint. But there are some key issues which make them a bit different. So first of all, the products, the investigation products, are going to have to comply with the ATMP specific GMP, good manufacturing practice, the guidelines on that. They're going to be particularly issues of quality of the human cells, quality of the medical devices. Let's not forget that in some products, because of combination products, how do you reconstitute the product? Then there's the safe conduct of the trial. How are you going to, you've got to provide detail on the risk, what is the risk and how are you going to handle it? Think about vectors. What about the risk minimization features or measures that you might employ, for example, into cytokine release syndrome? So important things there. Another factor that creeps in, a lot of these products are gene therapy medicinal products, and I will abbreviate whatever it was I was going to say that at least at the legal level, we're in a strange world whereby a genetically edited product isn't actually a gene therapy medicinal product. In fact, technically, it's not even an ATMP. But the regulations don't quite reflect the reality of genetic editing techniques. So it's a bit wonky, but more to the point when with a gene therapy medicinal product, and edited products are assumed to be gene therapy products quite reasonably. You have an extra layer of regulation, which is the GMO regulation, and the difficulty that then arises is that the process, certainly in the UK, is not too difficult because we have a contained approach. We have is that it's mostly contained use, but there are these two different systems, deliberate release, contained release, and they're different around Europe. Why does that matter? Because if you want to run a multi-center trial. So there is an issue over that which makes it more complicated. Now, in fact, I should just say that the UK, of course, going on its own merry way, is reviewing GMO regulation, and I believe that there will be, I hope at least, provision to make that side of life easier than it is at the moment. But perhaps more importantly, the EU is as well. We might hear some more on that in April. So there are some real differences. Right, what are some basic differences about impact things that occur to me to sort of tee up Tom? Well, one, I think we've heard quite a lot about accelerated access, conditional licenses, adaptive pathways. So I don't think I can manage that in the time, but let's just mention the innovative licensing and access pathway as well, which I don't know too much about. The homebrew issue is going to be an important one. And let's just put it simply. It's an exception to a rule which requires you to have the marketing authorization. And that authorization is important because not the least effective having a protracted development time, as you're losing patent time anyway. So you're losing that everything. So if you were hoping to rest everything on the patent, you may be making a mistake. You're going to have to rely on the data, orphan status, so you might have up to 10 years there, for example. But that's an MA. An exclusion, such as the hospital use exemption, obviously changes the whole model. Why would you invest if in fact you didn't need to? By and large, developers haven't needed to. The basic reason is this. When the 2007 ATMP regulation came in, we all thought that it was going to be bags of Geron labeled cells whizzing around the world. And they were allogeneic cells, they weren't autologous. The world's turned out differently at the moment. They're the vast majority of products are autologous cells. How do you put your own cells on a market? So the model doesn't quite work. So we have this strange thing. But the reason that there is an exclusion under existing medicines regulation, Specials Exemption, which has also tended to threaten this exclusivity, which hangs on to marketing authorization. So does it matter? Yes, it obviously matters if you don't get your kickback. It may challenge the very model of regulatory framework, which is geared to products, whereas the old mantra, of course, in regenerative medicine is the product is the process. In this case, a service, so that might do it. I think a thing that Dom and I particularly want to explore is the idea of what do you do if you've already got one? How do you reuse the data you've already got? So I think I'm going to shut up with those thoughts. Maybe we can look at that question of extension in our discussion. But I'd like to, if I may, Dom, turn over to you now. Is that okay?
Yeah, terrific. Thanks, Julian. Thanks, Kate. Yeah, I mean, from an investor point of view, these of regenerative medicine, advanced therapies, gene and cell therapies, are actually very interesting despite their regulatory challenges. And part of that is that they are newer products. And also they are a lot more complex products. There was a big shift when going from small molecules to antibodies. Antibodies were considered complex biologics, more complex than, for example, peptide drugs like an insulin. And now we're going to something like a gene therapy, for example, where you have a viral capsid and a expression cassette, some genetic material involved, which is like just on another level complicated. Or even living products like the CAR T cell therapies, where you have living cells that have been manipulated, which could be, Julian said, autologous or allogeneic. So it's on another level complicated. But the reason we like them is there are lots of challenges associated with that additional complexity. But the reason why we go to that length is the opportunity that these therapies have for the patients, that these are drugs that, and now they are approved examples of all of these categories that can actually address very significant unmet needs that, and a lot of these that have prior had often no treatment available. Last line, liquid tumor cancer patients that were very close to dying, or monogenic diseases that lead to, for example, blindness in the eye. And again, the only therapies available were reading aids, maybe a dog for the blind eventually down the line. And that's not really satisfying. And we live really in a time at the moment it's very exciting, where we have all these modalities available all of a sudden that really do make a difference. And I think in the coronavirus pandemic, we have it learned as well through the mRNA vaccines, where there's a much quicker way to develop vaccines, which will be potentially helpful if coronavirus happens to mutate and stick around and we need to all get vaccinated on a regular basis. So that's why we like it, and that's why also regulators like it. And we need to balance as an investor the risk benefit, and need to go first to those patients that have the potential to have the most benefit, like somebody who is likely going to die from their disease, or somebody who is doomed to go blind. And then the important bit is here just still to do the right thing to drive for excellence in these developments. Hence, there's sort of less concern around homebrew, because those will always be smaller data sets, usually local regional data sets, which not necessarily apply to larger populations. I think the commercial data set will usually be the stronger one, the better controlled one. And we make use of all these various, as Kate pointed out, frameworks that are there to engage with regulators early on, be it fast track, breakthrough, or ARMAT in the US, to name just a few, to get those regulatory interactions early on a higher level than you would get for just another small molecule drug, for example. And there's a reason why the regulators are happy to do that, is because there's more uncertainty around. They appreciate that you need to plan earlier, for example, which endpoints are we going to look at in our phase 2 study to then design the right phase 3 study, and to get that input early on to know, what are endpoints worth looking at, to still ultimately, as an investor, you want to optimize. As a competitive company, you want to optimize timelines, costs, risks, to stay competitive in all of these areas, and to have a commercial product in the end. And so these interactions are really sort of the key here, going after diseases that really have high unmet needs, and going with a technology that can really move that unmet need in a meaningful way, because ultimately the regulators need to take the decision, what is the risk benefit that I have seen? And given the always greater uncertainty you will have with these advanced therapies, that will always have initially smaller, shorter data sets, greater risks, so that needs to be explored. And that's why we need to follow up gene therapy patients for 15 years, for example. It's a long-term follow-up commitment on our companies to do that for a good reason, but that shouldn't mean we should wait to help these patients to preserve their sight, for example, in the eye with the approved product Luxturna, obviously. And so this is really how the investor looks at it, at all the opportunities and the challenges along the way. Is just the perfect environment to be in a small, nimble biotech environment to solve all these issues, probably better positioned actually, quite frankly, than a large pharma company, and greater competitiveness when looking from an investor point of view, given that sort of strategic example where a whole company focuses on 1 or 2 lead programs in a specific indication and really lives and dies by that, versus a large pharma company that maybe initially tries to sort of get into these spaces, but that's not where the main revenue drivers come from at the time. So for us, it's really opportunity, new modalities that do work, that help patients that are previously not helped, and all the extra effort, the extra risks, is just worth spending that time and working through that together with all the stakeholders, the patients, the regulators, to find the appropriate paths for these therapies, which needs to have the balance. In a very rare indication, you just can't get hundreds and thousands of patients of data, but you still need to find ways to make it a safe process that gets there to the patients at the right time. And so a lot of accelerated approvals, post-approval commitments here, so some elements that are used. And obviously these drugs work. I mean, the Avexis, the Zolgensma drug, obviously is close to a miracle. I remember seeing the data when they were first shown, of these children, walking around, playing with iPads of Pad 1. To me, it's really heartwarming, and so that's why it's worth doing it. And with that, I think I would really like to sort of answer a few questions as well, since we only have a few minutes left to be a little bit more interactive.
Can I pose a question actually briefly, which is that so many of these target conditions are really very, very niche. They're very small populations. How much of a challenge is it to you as investors that these are rare diseases? Yeah,
very good question, Julian. So there are 2 types of rare diseases, and one is the Zolgensmas of the world, the Luxturnas of this world, which are already very rare, but they're still large enough to be clearly a commercial opportunity, just based on the pre-existing pricing precedents, etc. But then there are actually hundreds of monogenic diseases as well, that are extremely rare, where you have just a handful of patients in any given country in a year, for example. And this is challenging right now to develop these drugs, because you still would have to provide a decent data set, and as you should. And the question is there, though, one day if you have, for example, now in the eye, you have a capsid and a promoter so that you can deliver to a certain cell type, express the gene on a certain level. How can we find a way to facilitate the development now of a drug for a patient population which is maybe 100 times smaller than the population that Luxturna addresses? And that's currently a completely uncharted territory. And it's a little bit of a challenge too, because regulators would want the company to start thinking about this and approach them with a solution. And until somebody has actually an idea that's worth starting to get going on, development, starting to interact with regulators, it's sort of a little bit of a standstill. But I think it would be a great shame if we not find a way to develop the drugs also for these very rare indications where the majority of them will probably work.
So slightly different. That's saying that the problem is one of having to go back and produce a whole new data set where in principle some of that data has already been produced. Why?
Especially around safety, yeah.
Yeah.
So the sweet spot is this common platform for lots and lots of sort of hub and spoke for lots of different monogenic conditions, say.
Yeah, I think once you have sort of established good safety, the one outstanding issue is a lot of the in vivo gene therapies is the inability at the moment to redose. So there's another loss. So if you then give somebody something where you're not sure about the efficacy, you might prevent that patient from receiving it may be future efficacious treatment ever again. But other than that, once you have sort of safety established, There just must be a way to create the appropriate data in a more expedited way, in a more post-approval scenario.
I think there is, to an extent, that you can reduce the data required, and Kate may have something to say about this, for example, in relation to quality. Right? A well-controlled process already, a lot of the process knowledge already exists, but then as Dominic, as you were saying, if it's different indication, different sort of scale, the strength is going to come in and so on. One thing Kate had a review on is this, you might need a new cell bank. Is that right?
Sorry, say that again.
So if you were to have a common platform, say, and you reapply your platform to another condition, how about having a new cell bank? It's not presumably sharing the same cell bank between products is not a great idea.
Yeah, and I think that there are ways that things like that can be done. And I think when we're moving, and we've seen it in large areas across quality, that we're now moving into that, we're really risk-led now as an industry, and that's going to be the key part of being able to do all the things that we want to do, like, where you've got safety data already in a patient population that you've, I'm thinking that there's a disease that comes up in the media all the time here is Batten's disease. And, the parents of those children really want a therapy, but there's so few patients that it's difficult to get something, up and running for that. But if you know that is actually safe, and you've got the data for that, and you can show that there's no risk to using that same data to go forward in a study and to use that, and then to start at the next phase. Again, there should be a way that you can use risk to show that you're actually, again, that benefit outweighs any risks that you might identify and then mitigate. I think some of it, again, is around the communication of people being comfortable to go with these proposals to the regulators and say, this is what we can do, and this is how we can help, but this is what we need from you. Now, we'll do the work and we'll give you the presentation. All you need to do is say whether you can agree with it or not. So I think it's going to take a lot of bravery on the behalf of some of the companies to do that.
May I just, sorry, just to interrupt, sorry. There was one question that came to me direct. So I know because we're just over time, I was keen to see that passed on. So thanks for everyone for obviously staying with us, and it's a great discussion so far. So the question was, what are the most important things to demonstrate in a preclinical package for advanced therapeutics that can help add value and mitigate risks early on? So Kate, did you want to take that first, and then by all means?
I might leave that over to Dominic or Julian if that's okay.
No problem. Yep.
And a chance to talk.
The most important in a pre.
Okay, sorry, I switched my microphone off. So yeah, I mean, that is a great question, because of course I when I was talking, I crashed straight into, well, you begin in human trials in phase 2A, but I mean, it's an area which certainly as a lawyer, I don't get much to deal with, but it's not to say that we're not conscious of it and don't look at it as an issue. So I think just thinking about this in this preclinical stage, so you've got the pharmacology data, that's key. I mean, shall I just run through what those data pack requirements are so that everyone's familiar with what they are in that early stage? So we're all familiar with that. So you've got, first of all, you've got your non-clinical side, preclinical side, and which we're talking about. So that's going to be pharmacokinetic, pharmacodynamic testing, first off. But I think the question here is, how does it differentiate? Because there are questions about animal models and how valuable are animal models in this context. Organoid models, for example, might be more appropriate. Who knows? So it is a difficult area, but it's not one which I deal with so directly.
Yeah, I would say it's very specific to the exact problem you're trying to solve, essentially. So if you take the example of a gene therapy, we know we can deliver AAV capsids to the human body in various compartments and see expression of genes. The question is usually, do you get enough in? if you want to treat, for example, the brain for certain disorders, that's a very tough biodistribution challenge, and the key data there would be showing that from at least from a sort of probability of success and investing point of view. Whether that's the regulator's focus, it's a different question. Their focus will always be on safety, rightfully so.
I mean, I just bring in, there's one thing to bring in there that I think I mentioned, biodistribution. So let's say we have a vectored product. Well, there's the vector, but also there's the gene. So where is it gene? That's the insertion point, issues of that sort that can be demonstrated in vitro. And of course, toxicity as well. So those are things which you need to do early on. They're standard, I'd say, but they're harder when you're, say, inserting a gene somewhere and checking its expression. And if it's a regulated gene, then it's more complicated still.
So we are just over time now. So I'm afraid I will sort of have to close the meeting there, but fantastic discussion. Thank you very much to Kate, Julian and Dominic for your time today. And I hope you've enjoyed participating. And for all those on the line and attended, thank you for joining us. And I'm very happy, for those who are not connected already, very happy to make introductions afterwards. I'll do a follow-up email with recording. And Kate, if I have permission to share your slides, but we can also discuss that afterwards, so no problem. Lovely, thank you.
Okay.
So with that, I'd just like to wish everyone a very pleasant afternoon, and thanks once again. Thank you.
Thank you very much.
Thank you, Alicia. Thank you all.
Bye.
Bye all.
Bye.